All right, welcome everyone. We're going to go ahead and get started. I want to thank everyone for joining us today. And let me do quick introductions here for our two presenters. First, we have Troy Fugate, who is the Vice President of Compliance Insight. Troy has been leading a team of industry professionals and former FDA inspectors for over 23 years and has extensive regulatory compliance, experience in regulatory compliance, regulations, and regulatory requirements. So welcome, Troy. We're grateful to have you here today and helping lead this discussion. Joining Troy today is Rob Perry, who is the Chief Scientific Officer of GeminiBio. Rob comes with over 20 years experience in cell therapy manufacturing, expertise in facility design, technology transfers, and regulatory compliance. He also has extensive experience in GMP manufacturing, logistics, and regulatory compliance for cell therapy industry. So welcome, Rob. We're grateful for both of you to be here. And with that, we can jump into the presentation.
All right, thanks, Kim. And hopefully at the end of this, you'll have a little bit better understanding of some of these critical materials that are used in cell therapy and some of the compliance and manufacturing requirements that are needed as you continue through your development process. So starting out is a little bit more of a background is what are cell therapies? Cell therapies are therapies which take viable cells. They're injected into patients or grafted, implanted to effectuate a medicinal effect. So if you think back to when the first true cell therapy was started, and a lot of people don't think about bone marrow as being a cell therapy, but true enough, it was. 1956 was the first successful bone marrow transplant. In 1986, it was the first TIL or tumor infiltrating lymphocytes were used to treat the cancers. In 2002, the first CAR T was developed. And then getting more into the current history, 2010 was Talon. And then 2017, CRISPR CARs were created. Next slide. So if you think about where we sit today, in 2024, there were 36 cell therapies approved by the FDA, with 9 cells for cell and gene therapies being approved. From 2021 to 2023, more than 400 cell therapy trials were conducted in the United States, including between 12 and 23 phase 3 trials. So if you think about where we're at in the development cycle, there's a lot coming on, and you can see that in the kind of the history and the effectiveness of the CAR Ts. The treatment modalities include CAR Ts, CAR NKs, gene editing, and tissue engineering, as well as TIL. If you look at the success rates for cell therapies in phase 1, where you're looking at truly safety, it's about 70 to 80%. When you get into phase 2, when you start looking at efficacy, those percentages drop down to 30 to 50%. And then in phase 3, when you're looking at statistical relevance, you're looking at about 50%. So looking at the numbers of 400 and you see it going through there, there's a lot to be excited about in the cell therapy world as the next decade kind of advances. Next slide. So looking at manufacturing of cell and gene therapies, if you take at the autologous cell therapy manipulations, what they're doing is they're taking the cells out of the patient, they're doing some type of manipulation, and then putting the cells back into the patient. That could be just immediately after manipulation. It could be that they grow the cells in the case of CAR Ts. When you're looking at allogeneic cell therapy manipulations, those are things where you create cell banks. Cell banks are well characterized. They're sitting in a freezer. When somebody has a certain disease state, you can thaw them out and then inject them into the patients. And then looking at gene editing, something like Talon, where you introduce the editing components into the cell using techniques like electroporation or viral vectors. The editing components create the breaks in the DNA. And then once the DNA is isolated within the nucleus, then the cell goes through a repair mechanism and basically puts that piece of DNA back into its DNA, and therefore getting the effect of whatever the DNA construct that you have on that vector. Next slide. So looking at different types of risk, you have a safety risk related to cell therapies. Does the product impart some type of safety concern to the recipient? This also includes all material and products that come in contact with the manufacturing process. It could be things like tubing. It could be the reagents that you use, and we'll be talking about one of those in a few minutes. It could be really anything, or really in the process, we're allowing some type of contaminant to be introduced into the process. Are your facilities up to snuff? Are they creating a potential contamination source? Or is it a risk to be able to manufacture in a grade C environment when you should be manufacturing in a grade A environment? Looking at the regulatory and compliance aspects, are all the materials that you're using compliant to the different regions of which you're planning on using the cell therapy product? And that goes for not only domestically here in the United States, but also internationally in Europe, Japan, or other parts within the world. And then the manufacturing process. Here we go. Manufacturing materials and personnel and facilities. So are the manufacturer of those reagents, are they using single-use materials like tubings and bags, or do they have some type of cleaning process where they've done a validation? If they haven't, is that going to impart risk into the product itself? What kind of testing is done on the raw materials before you actually use it in the manufacturing process? What are the facility flows that you have in the facility itself? Are you getting a potential of cross-contamination because you have material flows being crossed? How are the personnel trained? Are they undergoing some type of media fill process? Are they doing aseptic processing to show that they're qualified to be able to do the processes of which those reagents are being manufactured? And looking at the materials like human AB serum, are they collected at FDA registered or licensed facility? And is the adventitious agent testing using FDA approved kits? Is the equipment also FDA approved and using FDA materials? Next slide. So let's take a longer look at human AB serum. So human AB serum is used in the culturing of cell therapies by providing nutrients to the T cells as they expand. AB serum is used so that there's no antibodies to the blood antigens in the serum. Male donors are preferable because they have a lack of MHC antibodies, which could be present in females. And then looking at human AB serum, two types of serum. There's off the clot and converted plasma. So let's talk a little bit about off the clot. So in off the clot, a unit of blood is drawn, just like if you go to the Red Cross and you donate a pint of serum or a pint of blood, and then it's collected with the absence of anticoagulants like sodium citrate or EDTA. The blood's allowed to clot naturally, and then the liquid portion is removed and the clot stays back. After it's extracted and those two are separated, the serum is frozen, and then the site will do adventitious agent testing. Processing a batch of off the clot involves pooling donations, filtering through down to a 0.1 micron filter before the final fill and freeze. One of the drawbacks about off the clot, and it's something that we're trying to be very vocal on so that people, as they expand into their development process into phase 2 and phase 3, is that donors are allowed to donate off the clot material every 8 weeks, which means you can only get material from that donor 6 times a year, like you would with a pint of blood. So it's something to be aware of when you're looking at the indication that you're targeting and trying to set up your supply chain. Next slide. So let's talk a little bit about converted plasma. So the plasma is removed from the donor using the plasmapheresis device that has been approved by the FDA at a registered or licensed donation collection center. The plasma is then frozen and tested for adventitious agents, just like off the clot is. Then the converted plasma is pooled and clotted using some type of artificial thrombin. Now, there's a number of different thrombins that are out there when you're looking at converted plasma. It could be bovine thrombin, it could be native human thrombin, or there's also recombinant human thrombin. Material is processed for clarity and run through filters, again, down 0.1 micron before filling and freezing. But if you look at the potential for supply chain, donors for plasma can donate twice weekly, as long as there's 1 day between those. And that's because they're taking the liquid portion and the blood cells go back into the donor. Next slide. So human A/B testing, all adventitious agent testing should be performed using FDA approved and preferably CE marked kits when available. And the reason that CE marking is important is if you're going to be running clinical trials here in the United States, FDA approved kits are going to be the requirement when you go in and submit your IND. When you're going to expand into Europe, Europe's going to want to see that those kits are going to be CE marked. So having planning ahead of time, even when you're in preclinical or phase 1, if you plan and you have that already set up, you don't have to worry about it for the rest of the development cycle because you have it covered and you have some foresight thinking about it. Suppliers should have a look back program, and this is important for integrity of the material that you're using in human A/B serum. What look back program does is that it looks at these donors even after you have your collection and says, are they still clear of the adventitious agents that you've tested for? If they happen to get a positive, then the look back procedure allows the supplier of the human A/B serum to then look back, see what lots potentially are going to be affected, if they're going to be affected, and then if they have to, do a recall. And that's extremely important for the integrity of the clinical trials using that material itself. Donor testing combined with donor questionnaire, including travel questions, could help mitigate some of the qualifications of the donor. If you look at the testing panel that we at Gemini do, we have a couple of different types of serum. In the case of converted serum, we look at certain testing. It's the converted serum for research are the ones, the top 6, and the bottom one are the ones that we screen for. That's only meant for research. We do not screen for any of the others, but we do have 2 other different types of converted serum, and we test for the whole panel. What we want to do is we want to make sure that we're doing what we can do as a supplier of human A/B serum to show that the material that we're selling to our customers is has been tested and is as safe as much as we can make it based on the testing. And as new testing requirements come along, what we as the supplier, we also make sure that we add those tests to the testing profile itself. Next slide. So now we're going to talk about the regulatory aspects, and I'm going to turn it over to Troy to talk about these aspects of the human A/B serum. So Troy, take it away.
All right, great. Thanks, Rob. And I have to say, listening to you go through this, there's really fantastic to hear and understand this, and everybody listening to this understand the concepts. We go through the regulatory or the compliance aspects, the GMPs, so to say, with it, and it all comes down to control. The mechanisms you're talking about are all designed to look at, control the process or risk of the process, and how it goes into that finished drug product. So the foundation of this is going to be key. So establishing just what are the key regulatory frameworks with this? And I won't read all of these. Just understand beyond the typical CFR 210, 211, the GMP aspects of it, there are three key ones here I list out here. And so it's 21 CFR, all of these. It's part 640, it's part 610.40, and it's 1271. These are all designed to give the framework for the understanding of what is it that is needed to establish the framework, the solid foundation. And Rob, you talk about things looking at it. I look at these that you're speaking to as the foundation, the columns, the concrete, the steel that goes into it that is solid, and from there you can build up any type of framework that you want off of it, as long as you've got these things in control. So go ahead to the next slide, please. So there's a draft guidance with human AB serum, and this is presented here just so you can get some information. And don't worry, I'm not going to read this whole slide with you. It's a draft guidance on human and animal-derived materials in the manufacture of cell and gene therapy products. Basically, it goes through looking at these characteristics, the four highlighted sections here: a thorough materials characterization. What is the material that is being used? How do you know what it's supposed to do? A lot of times, from a regulatory perspective, I look at it and I ask people, what is this component supposed to do? And if they have an idea, sometimes it's very vague or very fuzzy. The second one here is probably one that is very critical, and I'd say it's one that people may perform, but they don't document. The third point here, but the risk assessment is understanding what is it that you have exposure to. What are the potential risks to the product, to the facility, to the patient? Say if you have mycoplasma and you look at that, or an adventitious agent that goes into it, the look back program absolutely is critical for these types of things. So it's understanding risk and mitigation of that risk. And then you get to the point of documentation, that third point there. Document what you're looking at. Why are you looking at it? Why did you make certain decisions? A lot of times we think things through, but we don't document the rationale, the thought, the concepts behind it. And then getting down to the last one there, the transparency and the record retention. The thing that I look at with this, if you look at the relationship here, Rob and I know each other, and we've developed a relationship looking at a process making this material and the compliance aspects that are needed with it. That is true transparency. And it's fully looking at what is it that we can do to make things better? What's the risk associated? What can we do? And to implement those things. So go ahead and to the next slide, please. So FDA summary of this. FDA advocates very heavily on patient safety, right? That they will look at this and say, patient safety, that's the certainly the core value of the FDA. They look at patient safety. That is their drive. So looking at this thing, the FDA expects human AB serum suppliers to have the appropriate GMPs implemented. I understand what I'm saying here with this is there's an expectation to have an appropriate level of controls. Take out the word GMP, those three letters, and put in the word control. So if you read that, suppliers are expected to have the appropriate controls in place, implemented with traceable donor history. Who are they? Exactly what Rob was talking about. Where have you gone? What have you done? documented eligibility screening, the viral safety testing, those panels that you go through and look at what could potentially be there. And it's always being updated. One thing I always appreciate, Rob, you put in there that as things are evaluated and other things come up in the future, you will add to that screening panel. So understand that last point on this left-hand column, even if the serum is used upstream of the final product formulation, they expect these things to be in place. The second bullet point here, the clinical stage companies have faced significant issues with the FDA because they failed to apply even a marginal level of rigor or decorum to non-critical reagents. They look at this as it is something you buy off the shelf. It is not a plug and play. It is something that you have to understand what is the functionality of this critical component in the process. And it is to take that rigor and look at it. So there's an increased scrutiny by the agency when it comes to ancillary materials. And that's going to play out in your future, either right now, you get into pre-IND, you get into IND, and certainly as you get into the BLA. And last point here is certainly 100% the FDA's drive with this is be proactive, not reactive. Be proactive in designing your trials and your process so that you understand what's going on. And if there are changes, that you can control those things, that the changes don't occur later in a development cycle farther down the line. They want you to have this mapped out and have it in a state of control. A key term I've used here, and Rob, you've heard me say this, is intent. You've got to have the intent to control your process and your material with the intent to make a product that is intended to have patient safety as the center core of that material and that process. Go ahead to the next slide. So EU regulations. Obviously, the US is a key component that I look at, but I also look at EU regulations. So there are various things with the EU that are the same but different than the US. So I understand there are some directives here that are listed here, but the key expectations include a couple of points here, right? 3 points. Use of plasma from limited donor pools. What they don't want to do is have a pooling process from a large mass source of donors. Second one here, full traceability of the collection and the testing. Traceability back to the patient. Traceability from the patient all the way through to the testing and the material that came out. It is basically a chain of custody concept. Someone comes in, where does it go, and where does it end up in this human AB serum? And thirdly here, documentation. Documentation showing that material collected from something, whatever it is not commingled with other things. This is the contamination, cross-contamination concept. All of these things come into place with understanding the EU regulations. And I could speak probably for about 2 hours on the regulations, but this is a real quick overview of things you should have in place. And certainly Rob and I have spoken about this, and part of the white paper is looking at this, and we discussed that at length in it. So go ahead to the next slide, please. So global regulatory considerations, right? The criteria for this and selecting and qualifying the human serum that comes out of it. So the criteria, if you go down on this, such as collecting or collection country, and US, its source materials often preferred due to existing infrastructure, FDA oversight, seems pretty simple with that. FDA registered collection centers ensures traceability. The donor travel history, as Rob mentioned earlier, Where have you gone? What have you done? The donor pool size, the gamma radiation, that's a great one here, mitigates viral contamination, but may impact the serum performance and may require revalidation as a concept if you do that. ADAG testing, advantageous agents, routine screening, the heat and activation, certainly one of those parameters that may inactivate or complement problems, but does not ensure sterility, certainly doesn't ensure sterility, and or eliminate the viral risk. And then the serotype and demographics, just the human AB serum is often preferred, and that is basically an understanding to reduce variability due to hormones. That it's just a basic fundamental of nature. Go on to the next slide, please. So, going in, the lessons learned working with regulatory agencies. There are two stories laid out here. One is a success story, right? A cell therapy firm preparing its first inspection for something they put together. They preemptively, understand what I'm saying here is they preemptively submitted information to the FDA. They were going out looking at the things that we're talking about here, saying, we are doing this ahead of schedule. We're looking at it. We've got this mapped out with the intent of having something that's in a state of control. The cautionary tale from this, there's absolutely many cases of this. There's another firm submitted its serum sourcing to a third party. They went out and did this work for them, but they failed to audit their documentation systems. Basically, it's a fundamental concept we always talk about. A chain is only as strong as its weakest link. If you look at the material, Rob, you're supplying to the people who are online that receive this material and may use it in a cell therapy process, the weakest link is the total strength of their entire chain of the process. So this second company ran into some delays and certainly some significant milestones that were missed. Bottom line with this, don't treat human serum like just another reagent. It is not just another reagent. This is critical, mainly due to its functionality and its sourcing and the risk associated with that material that may be collected. So summary with this, treat human serum as a regulated input. Very important. Validate your suppliers, qualify them, look at them, request that documentation, look at what they're doing, talk to them, treat them as a partner in this, and then engage the regulatory agencies early and have justifications, have it well thought out, map out a plan. And again, I tell you, I've always said this with Rob is when you deal with Rob, you certainly get the clarity, you get the confidence that the things that are being done are done correctly. So go on to the next slide, please. And Rob, I think this is where I hand this back to you to take it away.
Well, thanks, Troy. And again, we can't articulate enough. If you are planning on running clinical trials, a lot of times having the foresight and being able to plan for trials going outside the boundaries of the United States and others, it happens so frequently that if you plan for it ahead of time, that it doesn't matter what location you're going to be running your clinical trials. So a critical material like this or others, having the foresight ahead of time can save you a lot of time and a lot of money. So a little bit about Gemini's bench to BLA solution. Our facilities are constructed to minimize potential for contamination. We use all single-use materials when possible. We are ISO 13485 certified, and we adhere to the 21 CFR 820 GMPs. Our operations routinely perform aseptic process simulations, which are required, and also our technicians perform media fills every 6 months, not every year. We have filed a DMF for our human AB serums, and it has been looked at by the FDA. So if you're planning on using our material and running a clinical trial, all you have to do is talk to your sales rep about getting a letter of authorization and cross-reference, and we can write one for you that you can submit with your IND paperwork. And then what we'll do is we'll be talking to the FDA if they have any questions about this, and we'll deal with those questions accordingly. One thing is making sure that we have a product line with limited donors, so companies that are going to the EU have a material that they can use for these clinical trials. We do have customers that are using it now in Europe and in the clinical trials. So the EMA has looked at it and they've given their blessing to be able to use it. And really for us, we're here to support the regulatory aspect of this critical material. So when filing the IND, if you get any questions, we're also here to support that all the way up to BLAs. Next slide. So if you look at the different product lines that we have, our 100-512 is our research grade. As I mentioned, it's the one with limited donor testing. Our 912 is our human AB serum that can be used in the United States, depending on how it's needed. You can use it in different areas around the world, but for Europe, which is looking for limited donor pool sizes, our 916 is created where we have no more than 16 donors, but we create a batch size that makes it commercially viable for the companies that are using that product so they don't have to continually screen over and over and over again. And then we also have our off the clot product, our 100-318 product. We talked about the differences between that, but sometimes off the clot is the thing that is going to work the best. We just caution the people that are using it. If you're using it for a big indication, that you really need to look at your supply chain to make sure that you're going to be covered downstream of that. Next slide. So looking at the manufacturing risk mitigation, for us, we offer the material in bottles and bags. The bags are for closed system processing. Our aseptic assurance system reduces the risk so that you can do tubing welding or connections to things like bioreactors when you're growing the cells up to again, mitigate the risk of potential contamination of the critical cells that are coming out of these donors or out of these patients. We do custom manufacturing. We can provide the human serum that meet certain unique fill sizes. We can put 62 mils into a bag, 150 mils into a bag. We can give you bottles with 100 mils. We can give you bottles with a liter. It depends on what you want. For us, it's helping you to mitigate the contamination risk. We also offer heating activation, which is free of charge in the service, and we do have a validated gamma radiation process. So we've talked with our radiation vendor, and we've gone through the process of taking a number of different viruses and showing the fold reduction that you have going through the viral inactivation process using gamma radiation. And we have that as part of our DMF. Next slide. So the summary of the capabilities, Gemini supplies human AB serum to over 100 customers now, from preclinical all the way through commercial. We do have companies that are using our human AB product in their commercial applications. We've submitted multiple letters of authorization for cross-referencing the DMF, and it has been reviewed, as I mentioned, with the FDA. The supplier is positioned to scale with customers. We've talked to our suppliers. We know that as more and more successes happen out there for cell therapies, we want to make sure that we're with them every step of the way. making sure that we're going to provide them everything that they need to get these critical and really life-changing therapies to these patients. We also make buffers, medias, high purity waters, and things that are ancillary to the human AB serum and growing of the cells. We can customize the human AB products to help streamline the cell therapy manufacturing workflows that the different customers have. And we can scale our processed liquids from 10 liters to 10,000 liters. It just all depends on what you're needing and where you're at in the development process. Next slide. And really for us, it's having the industry leading quality department to meet the customer's requirements. We do produce animal derived materials like human AB serum, and then we also have spatially separated building to produce our animal origin free materials. And really, it's an integrated quality approach to meet the requirements and the expectations of our customers. Next slide. And so with that, what we'll do is we'll go ahead and open up to question and answers. And also, if you click on the QR code here, we provided a free human AB serum assessment checklist so that when you're going out there and you're looking for vendors of human AB serum, the things that you should look for in the vendor as you're moving into clinical development and beyond, making sure that they're covering those. And if not, make sure that you ask them as to why or why not, to help you kind of streamline your compliance roles as you're filing for, again, INDs and ultimately for BLAs. And with that, I want to say thank you, and we'll open it for Q&A.
If anyone has any Q&A, you can put it in the Q&A or chat and we'll pick it up. Right now, I'm not seeing anything quite yet.
See what, Rob, while people are maybe pondering the questions to ask, one of the things you and I speak to frequently is the concept of cross-contamination controls, and the cleaning of the tanks, the piping, things like that. You have anything to add to that? I mean, I know it's quite a lengthy process.
Yeah, I mean, it definitely is when you're looking at the manufacturer. And one of the things we do, again, we have our human AB serum separate from our processed liquids. Human AB serum, we try and use all single-use materials. We also do validation or performance validation of our filters at the end of the process to show that the filters remained integral throughout the process, so that 0.1 micron filter is intact. Looking at processed liquids, we've done as I mentioned, the cleaning validations for the stainless steel tanks. And we also evaluate every buffer or solution that we make to ensure that our cleaning validation meets those requirements for that liquid. And if not, we would do another validation to cover that as well.
Great.
We do have a question. One of our participants is asking, what is the specific regulatory document from the EMA that specifies the limited donor pool?
I think that's the one that was referenced, right?
Yeah, I think it was referenced on there.
One
thing I would say to do, if you go to the white paper that was written, that would have that reference in there. And I don't have it off the top of my head. I know most people.
Yeah,
Go ahead, Rob. Do you have it?
Yeah, the directive that we mentioned in the presentation is Directive 2002/98/EC. If you search on limited donor pool sizes for human materials, it would also come up. They don't specifically state what that number is. The filers are really the people who are required to show why 16 or 10 or whatever it is. For us, it's 16 because the pie has also mentioned their limited donor pool sizes in Germany would be 16 or less.
Yeah.
Okay,
I
we
think
also
that
have another.
becomes the C in the CGMP, Rob, just as people are doing this. Some of this is still evolving, but I know 16 is certainly a number. Go ahead, Kim. I'm sorry.
No, you're fine. We do have another question here. The FDA will require identity testing for critical reagents for BLA submissions. What do you recommend for identity testing on the customer side?
Well, I I definitely say from that perspective, and if whatever the critical reagent is that you work with the supplier of that material, identify the testing that would be used to perform ID testing of it. Basically, it's to understand the reason why you do ID testing is to ensure that it is that material. So you have to identify the elements of that reagent that would be specific to that reagent alone. And it may be beyond the concept of, say, a chemical test. It may be a functional test, but work with the supplier and develop that. A lot of this is develop it, especially if it's something unique to you, and document why you're doing this, and put that in your your basic QC and raw material specification files. Rob, do you have anything to say on human AB? I would go to you and basically ask, what could I do?
Yeah, there's a couple of things all of these donors are tested for human AB and blood type, and then we also have a biochemistry panel that goes along with it, which helps to set up the profile as human. We're starting to get more questions, and if we can elaborate on that, we will. But right now, those are the things that we can take to the FDA and say, look, every donor is blood typed. We have a history of it, and then when we do our batches, we do the biochemistry analysis with it as well.
Okay, we have a couple more questions coming in. One here is, have any of your clients worked with Health Canada? If yes, do you know if Health Canada prefers the use of serum from a small donor pool like the EU does?
I know that, Rob. And do you know that?
Well, I know what they typically follow, which is typically EMA standards.
Yes,
And
yes,
so
exactly.
they typically will follow that. And because of that, you can look at it as limited donor pool sizes, or you're doing something to mitigate risk, gamma radiation or something else.
Yeah. And it would be to follow the EU regulations on it. The one thing I would say, as you're engaged in Health Canada discussions and an application there, is to engage with the regulatory authorities in Canada and discuss upfront with them as to what you're doing and what their expectations are.
Okay, looks like we have two more questions. This one is, the FDA is also beginning to request the size of donor pools. Is this information part of the DMF or the C of A's?
So the answer to that is both. So depending on what product line you actually get, we have our 912 product, which is more complex in its number of donors. But we have our 916 product that is limited to 16 donors or less. And how we do that is we take these donors and they give multiple donations over a period of time, and that's how we get one lot of 16 donors or less. What was the other half of the question?
Is it part of the DMF or the C of A's?
Yeah, it's part of the DMF as well. We submitted the DMF for the three products that we have, the 100-912, the 100-916, and the 100-318. So all three of those uh, are covered under the DMF. The other thing about the DMF is it has history on lots. So we've been producing these materials over a period of years. And so the FDA can look at the DMF and say, how consistent is the manufacturing over time?
Okay, and we have another question here. This one says, we are using 2.5% human serum albumin in our manufacturing process. Is it possible to replace it with human AB serum? It's a three-parter. How would cost-wise will they compare? And do you provide albumin content in your C of A's?
So the is it possible to change? It's always possible to change. If you already have a filing, an IND, you need to talk to the authorities about the change, explain why you're doing it, and make sure that you have good sound rationale. What the FDA is, as Troy had mentioned, they're an advocacy group for the patients. They're the ones who speak up for the patients because patients can't. And so what they care about mostly is making sure that everything that's going into the patient or touches the product going into the patient is going to be safe. As for cost, I'd say talk to your Gemini sales rep and they'll be able to talk to you about cost. What was the third part?
And do we provide albumin content in our C of A's?
We do total protein. And if you look at albumin is 40 to 50% of the human AB serum. So you can kind of reference it to there, but we have total protein as part of that. And that's on the C of A as well as a number of biochemistry tests.
Yeah, Kim, one thing I would probably say to add to that first part, can they change it? The question I would have back is, where are you in the process? If you're early stage with it in development, it would be easier to change, and the farther along you get, the more significant of a change it would be.
Excellent. Those are all the questions we have for today. So we do want to thank everyone for attending. We will have this recording available online shortly, so we can always reference back to that. And we did reference the white paper in the chat comments for you, and the QR code for the assessment checklist will be available as well as in our follow-up. So we do want to thank everyone today. Rob and Troy, thank you both very much for your expertise, and I'll let you just do some quick closing comments.
Troy, you want to go first?
Sure, why not? Why not? Well, thank you very much, and thanks everyone for your time. I could just say from the perspective of FDA or EU, Health Canada, Japan, it doesn't matter. Think of your process as to control. And how do you control it? What are the risks? And then control those risks. And have the intent to develop a process that is controlled but able to be pivoted. As Rob, you said, one of the critical things, everyone planned on doing clinical trials in the US, and then when things shifted, money became an issue. All of a sudden, it was, oh, we're going to do clinical trials in Europe, and everyone was scrambling to get those CE marks or get the data together for various things. So have a well thought out plan as best you can, and then just stick to it.
Yeah, and I'll kind of reiterate something that you said, Troy, is that talk with the FDA or talk to the regulators because they're going to be an advocate. They're not scary people. They're not people to be feared, but they're there to help you in the ways that can help you out most. I know that we were able to, in my history, we were able to ask them about some information about a safety study, and they told us, well, you should probably take these 3 extra tissues that you don't have on your list, and that potentially saved us from having to run a second study, which would have cost us anywhere from $750,000 to $1 million. So by asking a simple question or simple questions to the regulators, they were able to help us out, and they're there to help you out. So, reach out to them and make sure that, you ask the questions, because you need to be informed as you go into your development cycle.
Great. Thank you, everyone. We're going to go ahead and end the event, and we appreciate everyone's participation today.
Thank you. Thank you very
Thank
much.
you.